So today it's my great joy to introduce Laura Podzikowski. She's known to many of you here, but I'm going to give you a proper introduction because you deserve it. So as many of you know, she received her PhD from our very own Dr. James Beaver's lab in 2023. She defended her dissertation with honors. Prior to that, she had her master's degree from the University of Michigan in conservation ecology, and she received that in 2014. And in 2009, she received her Bachelor of Arts from Kalamazoo College, also in Michigan. She majored in biology. It's worth mentioning her minors, which I love, environmental studies and German literature and language. It's wonderful to be well-rounded and to be surrounded by well-rounded people. Since her graduation with her PhD in 2023, she has served as a postdoctoral researcher in my lab. I should mention that her PhD was in part funded by a pre-doctoral fellowship from a proposal that Laura wrote that was funding from the USDA NIFA program. And for her postdoc, she wrote kind of a follow-up proposal that really springboarded off some of the interesting results that she found out of the Dimensions project for her dissertation. And that was funded by a USDA NIFA fellowship also, but a postdoctoral fellowship. That was working in my lab, and now she's on a one-year postdoc also in my lab using NSF the Earth Sciences program money to further explore some of the interesting findings from the USDA pre-doctoral fellowship and postdoctoral. So today, Laura is going to tell us two stories about soil organic carbon, and I'm excited to hear them. Please join me in welcoming Laura.
Hi. Today I'm going to talk about the influence of altered climate, soil structure and shear and roots on soil organic carbon dynamics in terrestrial ecosystems. Oh, I got to click. There we go. So since the Industrial Revolution, we know that human activities have been the dominant force governing global systems, and we know they're changing relatively rapidly. In this, they've outlined a bunch of these global change drivers, including climate change, biodiversity loss, and land use change. And you can see since 2009, we have been slowly crossing more and more boundaries for what is the safe operating space of Earth. The safe operating space is green, and anything that's beyond that means that we are altering that system at a rate and a scale that is not sustainable. So we know little about how these rapidly changing global forces will influence, such as altered precipitation, land use change, and biodiversity loss, will influence processes that we think tend to occur on a longer time scale, such as soil development and soil carbon generation. And this is one of the puzzles that my research studies. And today I will explore this theme with two stories. For those who do not know me, I'm an ecosystem ecologist, and I study global environmental change and plant biodiversity loss and how that affects terrestrial communities and ecosystems. And today we're going to focus on the ecosystem component for this talk, which will have two parts, and then I'll give you some ideas of some of the future directions in my research. So in the first part, we'll explore the influence of climate variability on soil development at the scale of the contiguous US. It will additionally explore the implications of that relationship and what they might be for carbon distributions and accumulations within soils. And then the second part of the talk will explore the relationship between plant biodiversity and soil carbon generation or regeneration, specifically in grassland ecosystems. Okay, so let's start with the first part. So it's well established that mean annual precipitation mediates the development of soils. And here we depict that as we move from a desert ecosystem to a tropical system. If you focus on this lower layer where the soil meets the bedrock, you can tell as you move as you increase in precipitation, it's getting deeper and thinner, whereas soils are being weathered. And we know that we define soil profiles by the movement of particles, right? So we know soils have layers, and they can be defined from the top all the way down to the bedrock. And it's often defined by the movement, the loss, and then the accumulation of different particles, and often clay, in part because clay is the smallest and most degraded particle, and so it dissolves well in water and infiltrates down the profile. But I just want to note that while it's often generalized that these horizons are defined by clay, so the horizon in the last slide, I always point it in the wrong direction. These horizons are here, and this is specifically, I said it was soil profile is defined by loss and accumulation. V horizon is this lower one where we accumulate materials like clay. And I just want to note that while it's often generalized that V horizons are defined by clay, that's not always the case, like in this desert soil. So for the purpose of this talk, we'll actually define soil development as regeneration of a T horizon. T stands for translocation, and it's defined by this distinct band of clay within the soil. So it actually is a measure of the movement of clay down a soil profile. And while we'd expect our VT horizons to deepen and thicken with precipitation, it's less clear how altered precipitation will influence the process of soil development. As global temperatures are expected to rise, precipitation is expected to change, just generally the amount, but also it's expected to become more variable with future warming. And this is occurring on a relatively rapid time scale. We don't really know how this might affect processes that we tend to think of as occurring on longer time scales like soil development. So we're going to study that, and we're going to do so in this PEDS data set. We're doing so in this PEDS data set because it's a large data set. It addresses, or it characterizes soil. I'm going to call them pedons. I'll define it in a second. It assesses about 20,000 or just over 20,000 pedons across the contiguous U.S. What that allows us to do is have a wide range of climate conditions and a wide range of soil developments for us to address this question across these natural systems. So soil pedons are these large 3D structures of soil. They extend deep into the soil in theory to the bedrock, but often as deep as it's safe to dig. And then they're usually big enough in scale, 1 by 1 meter at least in size, so that we can characterize the large structures of soil. So not just those fine distributions of particles, but also how these are all aggregating and building into large structures. This data set was compiled by Dan Hermes and Oesta, and it is compiled from the USDA NRCS soil survey data set, which characterized soil pits over the last 100 years. And typically one of the features of this data set that's also worth mentioning is that it not only does it have a wide scale and lots of points, but it also was able to quantitatively describe things in soils that are typically described qualitatively, which allows us to do some statistical tests that are a little bit more robust to address these questions. So this model, I'm going to model soil development as the depth to BT and thickness of the BT layer. And I'm going to see how it responds to variation in precipitation. So variation in precipitation here will be defined by the coefficient of variation, which is essentially the standard deviation normalized by the mean. And I'm doing the coefficient of variation across mean annual precipitation. Those mean monthly precipitation, mean monthly precipitation, not mean annual. So I'm calculating the CV precipitation across these mean monthly precipitation values, which were collected for over 30 years. And so in this model, we'll have this depth to BT, which will increase as you go down the y-axis. CV precipitation will be less variable the further to the left you are on this y-axis. And what it would look like is that you would have relatively consistent mean monthly precipitation across the year. High variability is on the right side of the axis, and if you look, that profile would suggest that you're getting most of your precipitation at like a specific point in the year. And so when I see how depth to BT or this soil development is influenced by the variation in precipitation, you can see when variation is low, the depth to BT is deeper. And when we have high variation, it tends to get shallower. And then when we try to compare this with shrink swell capacity, which is essentially a way that we would imagine that precipitation would be exacerbated. So shrink swell capacity, if you have a house and a foundation around here, it can increase the flashiness with which precipitation moves through a soil profile. And by comparison, when that's low, we might expect greater percolation into the soil. And you can see there's an interaction between that relationship. So that relationship between climate variability and depth to BT is steeper when coal is high or shrink swell capacity is high. And essentially, there's no relationship between those two variables. Wind shrinks soil capacity as well. Went too far. And this corresponds with an increasing thickness. So when it's consistent, precipitation is consistent. This corresponds with an increasing thickness of the BT layers. And what happens is that relationship between the thickness of the BT layers and the coefficient of variation in precipitation gets even thinner when you have high variation in precipitation. So what this says overall is that consistency in precipitation is really key for developing soil BT layers, deepening them and also thickening them. And what we might expect as climate changes in the future, if we have a system that's moving from a mean annual or mean monthly precipitation graph that looks like this to becoming more variable and distributed less evenly throughout the year, you might expect BT horizons to get shallower and thinner with future climate. So what, if anything, does that mean for this development of the SOC, which tends to follow clay? So there are often multiple ways you can measure soil development or describe it. I described clay as one of the ways, but another way that we can describe it is with the development of soil carbon. And why are we interested in soil carbon? Well, soil carbon is the largest terrestrial carbon stock on Earth. It represents, I think, 75%-ish, between 70 and 80% of the total global carbon stock, and is approximately the same size as the atmospheric and vegetation stock globally. So we're really interested in how soils develop carbon and store them. Typically, the concentration of organic matter will decline as you move down a soil profile. So at the top, you tend to have a lot of organic matter produced by plants. As they decompose and get smaller, they infiltrate down soil profiles, usually dissolved in water. And the concentration decreases as you get deeper. But even though 53% of the carbon found in those top 3 centimeters of soils is very important, some of the the 81% of the carbon found in those deeper profiles, where the concentrations tend to be lower, often are older. So they tend to be over 50 years old, which means that deep soils, even though they represent a little bit less than half of the total carbon stock, are actually a really important reservoir for carbon that remains stored long term. And I think that was this is just here again to show you that, right? So you have the top 30 centimeter soils here and the deep soils here. And it's just important to note that even though the concentrations decline as you go down a profile, the stocks are still an important reservoir for deep carbon and represent about half of the total, or just under half of the total stock in soils. So how do we measure the distribution of carbon and transport? It's actually quite difficult to do within these data sets, in part because these are static measures taken at a single point. So one of the ways that we can measure this is or approximate it, we can develop a proxy for it, is to just graph the relationship between organic carbon and depth. We can fit a curve to it. It tends to decay exponentially. We can calculate what this K rate is, and this will help us describe the shape of this curve, right? So I guess in theory, you would think the more curved this is, the higher the K, the less transport that is occurring, and that might be true. It's also possible that the less steep that relationship is, the less carbon there is in the surface soils, which tends to happen in croplands, which are kind of a unique case. So in croplands, topsoils tend to be eroded and experience a great deal of soil respiration. As a result, they tend to have significantly lower concentrations of, significantly lower concentrations of SOC. And then as you move down the profile, their roots don't root very deep, so they tend to retain a significantly more SOC, a little bit deeper. But overall, what that means is homogenizing or straightening of that relationship. So the K value gets lower. And when we probe the data set, I can assess whether or not a system has experienced agricultural disturbance by looking for an AP horizon. And when that's present, in general, the K value is lower, so it's essentially a flatter relationship. And when it has not been plowed, that K value is higher, so it's a steeper decline. So using this K value, I can assess, or I can just briefly look and see how does climate variability influence the shape of that relationship and that decline. And what you can see is when there's high variability, you're more likely to have a low value of K. So you're more likely to have a more uniform distribution of organic carbon throughout the profile. When variability in climate is low, there's a higher an equal probability that you can get a higher low. So the higher low K value. So that shape of that curve could be more curved or it might be straight. So this does imply that climate variability does influence the distribution of carbon found in the profiles, and they tend to be more uniformly distributed across that distribution. But the second thing we're interested in is how much carbon actually accumulates in deeper layers, right? And for this, we can actually do a larger assessment. So one of the things to note about that K value is it takes a lot of data to calculate it. You need at least 5 points across the soil profile, and you need to hit a threshold for the goodness and fit for that curve. And so it actually removes a lot of points because soil organic carbon wasn't measured all that often, or that often in all of those 21,000 pets. In fact, it was less than 2,000 that I was able to calculate it and get a good threshold for it. So another way to measure soil carbon accumulation in deeper soil layers is to create some sort of metric to look at these deeper horizons, specifically the V horizon. But when we do that, we know that we have to account for the most obvious way that that carbon will vary in the V horizon, which is by its inputs. So we divide it by the SOC in the overlying horizon, so the A horizon, to get a metric of accumulation. So the higher this value, the more B there is, or the more carbon there is relative to what was in the A. And when we explore the relationship between variation in precipitation and carbon accumulation, I ended up using quantile regressions to assess it, in part because what is surprising about it, and I probably should have shown you this, is when it's all black, it looks like there's a negative relationship between the two. And when you actually fit a line to it, it's positive relationship. And when you fit quantile regressions, you can see that we can get positive and negative relationships depending on how much emphasis we put on parts of this distribution, right? So essentially this relationship is changing as we go up this SOC or carbon accumulation metrics, which to me implies that there's likely multiple drivers that are influencing the relationship between carbon accumulation and climate. And we're going to explore that. So I've already mentioned for you, oops, I went too far. So I've already mentioned for you a few of the potential drivers, right? I talked about cover. So one of the ways that we can assess it is by comparing crops, forested systems, and grasslands. I've already hinted that texture is a huge important factor that influences this, and climate as well. But the last thing I haven't mentioned is that the structure of soil can also affect the accumulation of carbon down in lower layers. In this somewhat cartoon example, you can see that when heads are like smaller within, so if you have a lot of small aggregates, right, they might form a lot of pores, but they might decrease the ability of water to infiltrate or flow through them. So they might actually decrease infiltration. Or if you have large structures within the soil, they might decrease the number of pores, but increase the space that water can flow through it. So the structure of the soil in the overlying horizon may also influence the infiltration or percolation down that soil profile. To test all of these metrics, so this is like a huge data set, right? And so we want to think a little bit before we just dive in and start graphing relationships. And all of these concepts that we're really interested in testing are multivariate. So one of the things I decided that might be the best way to approach it is to develop multivariate metrics to test this. So I took all of the predictors that I thought encompassed or could describe texture, structure, and climate, and I put them into PCAs. And then I'll use these as these PCA axes, both 1 and 2, as predictors of carbon accumulation. And you can see that these, I'm not going to interpret these for you just yet, we'll get to that as we go through the model, but you can see that I'm at least explaining more than 65% of the variation in the data. So these are reasonable hypotheses for my PCAs. And I'll interpret them as we go further, but just a hint, they can be interpreted. Okay, so to assess the relative importance of all of these potential predictors, I did variance partitioning, which is quite simple. Essentially, I modeled the carbon accumulation, so the SOC in the B over A, as a function of cover class, PCA 1 and 2 from every single one of those metrics, soil texture, structure, and climate, and then I also incorporated their interaction with cover class. And then, so this model overall explains about 29% of the variation of data, which is actually quite reasonable for a noisy, biologically driven data set. I guess I don't have to say that in a room of ecologists. I've presented this to geologists before, and I was always like, oh. Anyways. And then I ranked them in order of which ones were explaining the most to the least variation in the data, right? So first and foremost, cover class is explaining the most variation in the data. Second is texture. Third is structure. And actually last is climate. So let's walk through the relationships that we have expectations for first. This is a somewhat abstract exercise. So we want to make sure that what we're seeing makes sense. The first one is we already talked about how we can get a high value. So we can get, well, I guess we kind of said it within the distribution, right? But you can imagine that uniform distribution would create a high SOC in the V ratio for crop cover, in part because they deplete an SOC in that A horizon. We see that in this relationship. I show that to you again. And when you actually, one of the nice things about this data set is you can probe it to make sure that some of the ways you're interpreting your data are correct. And when we do, we actually do see significantly lower SOC in the A horizon and significantly higher SOC in the B horizon. So that's good. And then when I look at these relationships between climate variables and cover class, and I just highlight the crop ones, for both the cold to wet climate access, or wet to dry, excuse me, cold to hot, I don't know, cold to wet, just kidding. But you can see the relationship is flat, which is what we would expect for crop cover, right? Because we can select plants that are capable of growing across the climate gradient or that are going to do as well as we think they are. We can also irrigate. So we wouldn't necessarily expect croplands to have a relationship between carbon accumulation and climate. And when we look at the relationship between PCA axis 2 for the texture and carbon accumulation in the V layer, we can see it's actually higher when there's more clay and lower when there's less clay, and that might make sense, right? We expect SMC to correlate with clay. So having higher clay in the overlying layer was a better predictor for carbon accumulation. Okay, so then this last set of analyses, I just want to point out we're still kind of in the process of interpreting and analyzing these data. We're still working a little bit through this structure PCA. There's a few tweaks that we might make. So I'm hesitant to interpret all of it for you, but I want to show you one of the things that we're seeing, one of which is that structure is more important than climate, and this is very surprising because in soils, when you do a PCA on a soil with climate and texture variables, you expect them to be the first and second PCA axis. So it's surprising that the climate axis is actually explaining less variation in data than structure. And then what's interesting is they all have this interaction between climate, cover class. And what you can see is obviously the croplands differ, but in most of these as well, you can see there's significant differences between tree cover or forested cover and grasslands. And in general, the relationship between all of these axes and grasslands tends to be less steep, more flat, and it tends to be more steep in these tree covering, or in these forested systems. One of the things that this might hint at, although this is not a done deal, this paper is still in the works, but one of the things this might hint at is that there might be different mechanisms through which carbon is moving through the profiles, right? So maybe in these forested systems, it really is dissolving in water, and that is driving these relationships. maybe in these grassland systems, they're able to actually root into these B horizons, and as a result, direct inputs are driving the carbon accumulation in that B horizon. We still have some more work to do, and then we'll want to probe some of those questions as well, or those hypotheses. But overall, there are fundamental differences between grasslands and forest systems that seem to be elucidated from these analyses. So in summary, what did we find? Well, consistency in precipitation tends to lead to greater soil development. And in general, variation in precipitation alters how carbon is distributed with depth. And lastly, the overlying cover and overlying structure, so the structure and cover that's influencing these top horizons seems to be impacting carbon accumulation deeper in soils in ways that are unexpected. For instance, structure was more important than climate, and in ways that suggest there's fundamental differences between systems like grasslands and forests. So in the second part of the talk, right, I just hinted at the fact that forests and grasslands might have fundamental differences in the way they influence carbon accumulation in deeper profiles. For the next part of this talk, we're going to talk about how grasslands specifically generate carbon in their topsoils. And one of the ways that they can do this is through diversity. So first, I guess we have to step back and ask, why am I focusing on grasslands? Grasslands store about 34% of the terrestrial carbon stock, and then they're the third largest soil carbon stock on Earth, behind wetlands and boreal forests, with a greater carbon storage below ground than actually timber forests. So they're an important reservoir for carbon below ground. About 90% of the carbon that is stored in grasslands is stored either in their roots or in their soil organic carbon, and we know that diversity tends to increase SOC in topsoils, right? However, and we see that in this relationship with the overall patterns. However, that relationship is inconsistent. So when you can see when they break it out for just grasslands and just forests, most of the time it's positive, but that relationship is not significantly different from zero, right? So in some of these cases, it's not consistently increasing SOC. And we also know that in natural settings, increased diversity does not always equate with increased topsoil SOC, which kind of begs the question, what is driving this? Some people have hypothesized that functional diversity could be driving this relationship. Specifically, they're very interested in the combination of these C4 grasses and legumes. C4 grasses photosynthesize at a relatively rapid rate relative to C3 photosynthetic capacities, and legumes can form symbiotic relationships with nitrogen-fixing bacteria, which increases their access to nitrogen, which is often the limiting resource in terrestrial systems. And you can see that in general, the combination of those two in this study tended to lead to greater carbon storage over a 22-year time span. But when you take that idea and apply it to other systems, like agricultural system, it doesn't always pan out. So in this study, we have this intensive grain production, which is a C4 grass. They did a rotation with legumes and added manure, and then they compared that to SOC storage in these well-grazed grasslands. And technically, this middle system where they added legumes and manures did not have significantly greater carbon storage in comparison to these, or did not have it in comparison to this intensive grain system, right? So that begs the question, why? And what's interesting about grasslands is, unlike these boreal forests and wetlands, we don't have a good idea about why grasslands store so much carbon. So like in boreal forests, we know that it's cold and wet, and that slows the rate of decomposition of organic matter. In these wetlands, we know that they are covered in water, which decreases oxygen, and oxygen is a really great substrate for decomposition. It allows it to happen relatively fast. So it also slows decomposition, which allows for greater storage. So we know that suppressed respiratory loss might be one way that we can increase carbon storage. And I guess if you probe this system, you can see with this orange arrow, I've tried to highlight it. They call it the mineralization of a specific type of organic matter, but you can see it gets significantly bigger. It equates to respiration for all intents and purposes. So it could be that increased rates of respiration are what suppressed carbon storage in that system in comparison to the natural grasslands. But we don't really know. So let's think for a second and step back and think how can carbon be allocated in these two systems? And I'm going to use this cartoon comparing forests here with this tree and grasslands. In general, forested systems allocate about 60 to 85% of their net primary production. So that's the amount of carbon that they take in and incorporate into their biomass. They incorporate about 60 to 85% of that above ground. In contrast, in grassland systems, about 60% of their NPP is allocated below ground, right? But what's interesting is in these forested systems, when the trees senesce, almost all of that is respired off. It's generally considered to be a net neutral effect on the carbon balance. So that could suggest that respiration or the suppression of it might be going on below ground and driving this relationship. But to really assess that, we kind of have to think about the whole cycle of carbon and how it is both generated and also lost. This is a pretty classic ecosystem ecologist diagram. My pools or stocks will be in squares. A process or transformation will be indicated by all these arrows. This is not extensive by any means, so if anyone studies carbon, I'm sorry if I missed your process of transformation. Okay, so we know that respiration, one of the things I want to note, right, is that all these processes at each one of these steps, that is essentially decomposition of organic matter, right? So at every single one of these arrows, we can lose carbon to CO2. Some of that is dissolved in water and dissolved down at soil profiles and groundwater. Some of that's released out into the atmosphere as soil respiration. And then when we start thinking about these carbon pools and how they're generated, so a plant takes up carbon and incorporates it into its biomass up here. That leaf litter can decompose and end up directly contributing to SOC. A relatively small fraction of that actually does, it's less than 10%. Most of the carbon in grassland systems is allocated below ground. We know 60% of it is. And that is either reallocated to root biomass or root exudation. So root exudation is the way that plants try to interact with the soil rhizosphere and microbial community. A fraction of them can end up, these root exudates can end up as soil organic matter because they're small and they can get bound to particles. Some of them will interact with microbes and increase the microbial biomass in the soils. Microbes can act upon multiple different stocks of carbon, right? So they can produce, I call these EEAs. Microbes, in order to get their nutrition, they have to try to get it from the soil environment around them because they're not able to move, right? So they produce these extracellular enzymes that break down resources that are further away, and then they hopefully break down enough of it that it diffuses to them. Those things that they're putting out are called extracellular enzymes, and we measure their activity as a proxy for the amount that's being produced. And it can act on these soil organic carbon pools. It can also decompose root tissues and root residues, right? And when these microbial communities get large, they also are growing and dying and turning over. They produce both necromass, so dead materials, and they also produce residues. All of those from their turnover can end up as SOC. And the last stock I probably should have done first, but maybe it's the most obvious. Plants can allocate carbon to root biomass directly. When that root biomass senesces, it can decompose, and a good chunk of that could end up in the SOC layer. And I will say that there are a lot of complicated ways that you can measure SOC. A lot of the studies I've been talking about have measured just this bulk pool. You can also measure the amount that's associated to different size classes within the soil. You can also do different types of extractions to assess whether or not it's more or less soluble, and this EOC represents a more soluble form of SOC in total. And so that is part of what makes this such a difficult system to assess, right? This is a lot of things to measure. But guess what? I measured most of them, yay! In a specific experiment. So we'll be able to walk through some of these processes and analyze how they might influence SOC storage. So where I actually measured most of these things is in a biodiversity manipulation experiment. It's a full factorial manipulation of precipitation, plant richness, and community composition. We manipulate precipitation from these rainfall exclusion shelters, which exclude all of the rainfall. We collect it in these barrels, and then we reapply it at 50% and 150% of ambient growing season rainfall. So we have an elevated and reduced precipitation treatment. We have 18 species, and from every plot, we selected between 1 and 6 species to plant in those plots. So plant richness is the number of species in that plot, and we've manipulated that. And then the last thing that we've also manipulated is the community composition within that plot. And we did so because those 18 species were from 3 different families, and we selected the diverse mixtures, the 2 to 6 species mixtures from either multiple families or a single family. And here's an example where we have 3 species, echinacea, we have a dahlia in here, and we have a grass that's a multi-family 3 species mixture. This is a 6 species mixture of all grasses, which you'll have to trust me on, I know they look all the same. And then we have 2 species in here, a helianthus and a grass species, right? And we specifically chose these families to have different functional groups. So we're manipulating family diversity. That also coincides with a specific type of functional trait diversity. And we just so happen to manipulate some of those characteristics that we were talking about earlier in this talk. So we selected species from the legume family, and as I said, they form symbiotic relationships with endosine bacteria, so they tend to have relatively high tissue nitrogen content. I will depict them with a peanut. A peanut is not one of our study species, okay? It's just a representation that I would hope will resonate with you. The entire idea is that legumes are supposed to be more palatable and more nutritious because they have a high N content. Although peanuts are legumes, so, okay. Then we had grasses, and most of our grasses are C4 grasses. I think it's 4 out of 5 of them are. And those C4 grasses, as I said, can photosynthesize at a relatively fast rate, so they tend to have high tissue carbon content. I'm depicting them with a piece of kale. I know kale is a brassica, okay? But this is meant to be like what you would think of as being like eating grass. Something that would be less nutritious and lower on your list of things you would choose to eat. Okay, and then the last family that we manipulate were asters, which tend to be tall and deep rooting and produce these flowers. A sunflower is in the aster family, and we do have a species of sunflower in them. And so I will designate in red the legume family, mixtures with all legumes, in green, mixtures with all grasses, in yellow, mixtures with all the aster family, and then we mix all 3 of them in 2 to 6 species plots, and that's our multi-family mixtures, right? And then for some of these experiments, right, so when I measured certain things, I will tell you it's actually time intensive to do that. I measured root exudation and root decomposition and leaf litter decomposition. It's a lot of work to measure those things. So when I do that, I'm using focal species, and I happen to use the same, so all of these species are one of the species in our plots. I use the same species in every single one of my experiments. So for the legume family, I use Dahlia candida, white prairie clover. For the grass, I use panicum, switchgrass. And then for the aster family, I use silphium. Okay, so let's address this first question. Does SOC actually increase with diversity? Yes, it does. After 5 years after establishment, we saw a significant positive relationship between plant species richness and the carbon in the topsoil. And this is a pretty remarkable relationship because our soils were not relatively deplete in carbon. So it is kind of remarkable that we were able to see that so quickly. Okay, so then does that influence community composition? So in this diagram, I've calculated an effect size. So this positive effect size of SOC is the relationship between richness and SOC is significantly higher than 0. So we have a significant positive relationship, as you see here. I'm not interested in the relationship for all of these community composition metrics. I'm interested in whether or not they significantly differ from one another. For SOC, they don't, right? So we didn't see a significant effect of family composition on SOC accumulation. And actually, I'm going to tell you really quickly, the same was true for precipitation. So we were able to generate SOC in these communities with all of these different family types. and with high and low or elevated and reduced precipitation, right? And this is interesting because it's consistent with some, but not consistent with other studies that suggest multiple families may influence SOC. And I'm going to just go through and walk you guys through an exercise because I've measured all of these things. Even though we were able to generate SOC in all of these single family mixtures, I hypothesize that it's actually happening through different pathways. And I'm going to walk you through why. And I'm actually going to start with the aster family because they seem to get ignored, and I think they're playing an important role in the generation of soil carbon, so I'm starting with them first. When I measured this EOC pool, don't get afraid by this SEM analysis. It's all the same boxes, right? So we have above ground biomass here, here, soil respiration here, here, root biomass here and here, microbial biomass here and here, and then SOC, right? So it's all the same pool. And when I incorporate the density of asters into this relationship, you can see it can have a significant positive effect on the generation of EOC. EOC is a relatively soluble form of SOC, right? Where might it be coming from? Well, I think it might actually be coming from increased rates of root decomposition. I did a root decomposition experiment with our 3 target or focal species. And to do that, I essentially took their root material, put it in bags, buried it in the plots. I collected it after 91, 211, and 300 and some odd plus 1 year plus days after they were deployed. And then we can fit this, you've seen this before, an exponential decay curve to it. This measure of K relates to the rate at which it decomposes. The higher it is, the faster the litter is decomposing. And silphium, that aster, decomposed significantly higher than all of our other species. In fact, it decomposed almost within 200 days, but it took approximately less than a year for all of the silphium root litter to decompose. And this was really surprising because if you were to ask me, and as I wrote this paper, when I put out my hypotheses, I will hypothesize that the legume should decompose the fastest because it has the highest tissue nitrogen content. And that actually wasn't the case. So there's some other feature of silphium roots that are contributing to the decomposition rates, and that may be increasing the amount of EOC and contributing to increased SOC with diversity that we're seeing in the aster family. So what's up with legumes, right? So legumes are kind of the star of the SOC story. If you read the literature, everybody wants you to add legumes to your plots. How might they be generating SOC? Well, I think I found through a few different mechanisms, one of which is through root exudation. I did a root exudation experiment with the focal species again, because it's very hard to measure. And as you can see, with the presence of this legume, this dahlia, we had significantly higher root exudation rates in comparison to grasses and silphium. Again, this is surprising, and when I write this paper, I will hypothesize that I thought grasses would exudate at the highest rate, or would have the greatest amount of exudation because they had the highest rates of soil respiration. So I expected that to be happening through exudation. That doesn't seem to be the case. And in general, it tends to, when I increase the number of dahlia in a pot, exudation tends to increase, so it seems to be an additive effect. Similarly, legumes tend to enhance extracellular enzyme activities. So I have extracellular enzyme activities here for carbon acquiring, nitrogen acquiring, and phosphorus acquiring compounds, right? When I assess this in the plots and compare the aster, legume, multifamily, and grass communities, you can see that significantly higher extracellular enzyme activities for all of these acquiring enzymes in the legume community. And this is particularly surprising for this nitrogen acquiring enzyme. You would expect a lot of people talk about these legumes as having a fertilizing effect on these plots. If you were to fertilize these plots, you would expect this nitrogen acquiring enzyme activity to go down. That's not actually what we're seeing. And the last way that legumes might enhance soil organic carbon generation is through increased decomposition of other plant litter. So this is actually from my dissertation. I did a leaf litter decomposition study. I was decomposing switchgrass. It looked very similar to the root decomposition study. And when I analyzed the data in general, switchgrass tended to decompose really fast with itself. which people, or with other grasses, which people might describe as home field advantage, being exposed to communities that are adapted to decompose. But surprisingly, legumes also had a benefit, and they had a benefit that was comparable to the grasses as well. So essentially, legumes might enhance exudation. They might enhance the availability of these, or the EAs, which can they can decompose these materials, but they can also add to soil organic carbon. And they also can increase the rate at which decomposition occurs for other species. And I have evidence of that occurring in leaves. I haven't analyzed the root data for it, but it might be in that data as well. Okay, so then what role do grasses play in this, right? So that's the other star of this literature. It's mixing C4 grasses and legumes. Well, grasses were able to significantly increase. So this is the same diagram you saw before, but bigger. And they were significantly able to increase microbial biomass and root biomass, both of which are big pools that contribute to SOC. But they also increased soil moisture and the rate of soil respiration, which act upon that organic carbon to release it back into the atmosphere. So those are potentially balancing or counterbalancing factors. But one of the things to note is that their root litter actually did decompose shockingly slowly, right? So often they produce a lot of fine roots, and people would expect their litter to decompose rapidly. They actually had the slowest rates of decomposition. They were decomposing so slow it could take 2 to 3 years before their root litter actually turns over. In comparison, the silkworm is about 1 year. This dahlia would be just over 1 year. So that's relatively slow. And to explain why that's important, I'm going to show you this study from a forested system. They decomposed leaf litter in gray and root litter in this blue. Root litter decomposed slower than the leaf litter, but when they explored the amount of plant-derived carbon that ended up in soil carbon from either these roots or leaves, the leaves only contributed to the soil carbon for the first couple of years, whereas these slow decomposing roots were continuing to contribute to carbon for many years thereafter. So slow decomposing roots might be the gift that keeps on giving to soil organic carbon. So yes, grasses did increase microbial biomass. They did increase soil respiration, we can't ignore that. But they also increased the amount of slow decomposing litter, which may be contributing over the long run to the generation of SOC. And their increased microbial biomass pools might be contributing more to these microbial necromass and residue pools. I don't know that for sure because these, I think they're almost impossible to measure, but they're really hard to measure. We can have a conversation about that if you want to. So in general, let's summarize how plant community composition might influence the generation of carbon in grasslands. Asters can contribute to SOC because their roots decompose really quickly, so they can contribute to the EOC, relatively soluble pools that might actually end up percolating down soil profiles. So that's really important. These legumes enhanced root exudation and the extracellular enzyme activities, which might directly contribute. They might decompose, but they also, because there's more of them, might contribute to these SOC pools. They also stimulated the decomposition of other litter. And then grasses increased microbial biomass, which might increase these necromass or residue pools. They also contribute to slow decomposing litter, which might be contributing to this over the long run. So let's hypothesize what would happen if we were to combine all three of those types or archetypes, right? The hypothesized effect would look like this. If we flip between my original diagram and this, what you can see is almost every single one of these arrows that is contributing to SOC is getting bold. So essentially, they might be contributing to greater SOC because essentially they're like flooding the zone with carbon input. I'm sorry. Too soon? Is it too soon to make that joke? But essentially, they might be altering the amount of soil carbon. So this is my hypothesis, right? Rather than suppressing soil respiration, they might just be flooding the system with a diversity of different compounds that can contribute to SOC. Okay. And one of the things that I want to note is that the evidence does suggest that it may be dependent on traits of individual species or within a family. And I'm noting that because I've showed you examples where we've mixed these different types and that we don't actually generate greater SOC. And one of the things to note is I showed you an agricultural system. In that system, they specifically select for plants that don't allocate resources below ground. They also don't often interact with microbial communities quite as much. They might not produce as many extracellular enzymes or give as much carbon to fungal symbionts that can create more biomass and more necromass and residues. So I am hypothesizing that some of these plants that we're choosing these biodiversity manipulation experiments have these traits that are associated with them that aren't necessarily, that might be held or phylogenetically conserved across the family, but they might not be, right? It might be dependent on how easy or hard they are to establish as well. Okay, and so then the last treatment that we measured was precipitation. I'm going to go through this really quickly because it's kind of the same story for precipitation. The path that's generating SOC with diversity in precipitation treatments is probably differing. And the way I know that is because I analyzed these data in elevated and reduced precipitation. And reduced precipitation, the path between richness is acting on root biomass, which acts on microbial biomass, which contributes to SOC. So the hypothesis in this scenario is that roots are creating a lot of exudates to stimulate microbial communities in order to withstand these reduced or stressed conditions, which ultimately contribute to SOC. And when you look at my root exudation experiment, I did have significantly higher root exudation in my low water treatment. By contrast, in this elevated precipitation, we tend to see that root biomass is the strongest contributor to SOC, and it's weakly related to richness through above ground biomass. And I am hypothesizing that perhaps the way that is happening in these high precipitation treatments is because they tend to stimulate root decomposition relatively rapidly. So we had significantly faster root decomposition and elevated precipitation right here. I probably am standing in front of the camera, people. I'm sorry, right here. For this legume, and we also had slightly faster root decomposition for the silphium, which might be contributing to greater soil carbon in these higher carbon treatments, right? So essentially enhanced root exudation and the reduced precipitation might be contributing to the relationship between SOC and diversity and reduced precipitation and elevated precipitation. SOC might be generated in part because of increased rates of decomposition. Okay, so in summary, right, we did see that diversity increased SOC, that it could happen in all of these community composition types and precipitation treatments, but I hypothesized that it's happening through different mechanisms. The combined effect on soil carbon dynamics when you mix all these species in the diversity treatments might be that they're essentially just flooding the system with a bunch of, or a diversity of different carbon substrates generating SOC, and elevated, or in reduced precipitation, elevated exudation is generating SOC, and by contrast, decomposition seems to be the contributing factor in elevated precipitation. So even though diversity can generate SOC in both family and species, in both these community composition treatments and precipitation treatments, there is difference that in the way or the path that was likely generating it. Okay, so for future directions, obviously one of the most obvious is we want to try to explore some of those, or continue to explore some of those patterns we've been seeing in the SOC dynamics and see that over time as we watch these experiments, if those communities start to diverge, because you can hypothesize from those individual pathways that they would. Another potential future direction, well, it's not potential actually, I'm starting to work on it right now, is to then also explore how carbon generated by plant diversity percolates down soil profiles. Because I have this diversity manipulation, I can collect soils across the depth distribution. I can start to assess how plant communities and grasslands specifically what mechanism might be facilitating the percolation or the accumulation of SOC in deeper soils where it's more likely to be stored. Okay, so with that, I have a lot of people, thank you, all these people, who I will say that this has been a massive collaborative effort, so thanks specifically to everyone who helped with the PET data set. That entire first section couldn't happen if they didn't do that. I think people for my field work, or who helped specifically with my field work in the Billings lab, the KU field station staff, the Beaver Schultz lab, a lot of technicians who contributed to the maintenance of these biodiversity plots, which is a massive undertaking, and then of course the many different funding sources that have enabled me to be able to explore this for what is probably now 7 or 8 years. So with that, I'm done, and I'll take any questions. I think Sarah was first, behind you.
Oh, that was a really good talk. A lot of information. So your relationships between species diversity and soil organic carbon. Why do you think there's so much variation within a level of diversity in soil carbon?
Yeah, well, there's a lot of things. One of the things that we're not accounting for in that model is NPP, so the biomass, which could be accounting for a lot of that variation. Within each level, it's a full factorial manipulation, and we have species that we've selected that are both early successional and late successional. So within each of those levels, I showed you the results from these species that I've chosen for a lot of these experiments, and they were intended because they were somewhat late successional. We expected them to have effects, but I didn't necessarily choose like the annual aster that might not be influencing soil carbon, might be behaving more like an agricultural crop, right? So that could account for some of the variation, because these are full factorial manipulations, and those are equally represented in all of our Alberta diversity treatments. I think Jim's next. I'm sorry.
Okay.
Then Jude.
So I've been trying to think, because like in the first part of your talk, you're talking about variation in precipitation and variation and distribution of soil carbon. And I was trying to keep in mind the total amount of precipitation and the total amount of soil carbon through the whole thing, and it gets very confusing because you showed results that the coefficient of variance is negatively correlated with total precipitation. So those are right? So they were opposite, they went in opposite directions. So high coefficient of variation is lower on average precipitation levels according to your axis of your PCA axis showed them in opposite directions.
Yeah. I was wondering where I was like, I don't think I showed that figure.
Yeah. So
because they are actually they're correlated in a very non-linear, in a cone-shaped way.
Yeah,
and they're
so
positively
that's good.
correlated in a
So
cone-shaped
I guess I'm
way.
just wondering, why can't you look at them both? Why can't you simultaneously look at
Mean annual precipitation and
total precipitation and the coefficient of variation to predict the distribution of carbon, but then also simultaneously look at the total amount of carbon, like put them, it seems like you have two predictors, total amount of precipitation and the coefficient of variation of precipitation, but you should have two responses, the total amount of carbon, which you have measures, and then the variance from the high to the low in the soil structure.
yeah.
And it seems like they all matter, and you put them all, you could do it all next to each other in the whole paper.
Yeah, I could. I will say that in the first section, I hid that slide, so let's see if I can even show it. So I will say that in the first section, when I talk about soil development, not when I talk about carbon yet, right? I think I left this slide in here somewhere. I do account for unhide, presenter view. I do, these are my covariates in the model. I account for two other forms of soil development, two other ways we can measure it, and this is also a covariate. So I do account for it in the first half, or the first part of it. And then the second part, I felt like I was accounting for it by including it in the PCA. But I would love to hear your ideas about how to do it differently.
But when you include it as a covariate, but what is the, I guess just reporting it then, right? So you have it in your models.
Yes, I do.
And so but it might be good to report it simultaneously.
Yes, we definitely will, for sure.
Yeah.
Because it will Yes, it will definitely. It's one of the things that I always plot as well, just not for this talk.
Because, and also similar for total soil carbon, because if you talk more about total soil carbon in the first part of your talk, then it would connect better with the second part of your talk, which is all about, where you don't talk about stratification yet, you're just all about total soil carbon.
Right, right.
I have one other question. Jude's next.
Okay, Jude then. Great.
Thanks, Laura. That's really cool. And I hope I don't know if I understand this exactly right. If so, it's kind of complementary to Sarah's question a little bit. On your factorial design experiment, did SOC go up in every situation?
In every,
I know with your community, you pointed that out. It was like, oh, it went up and measured 5 years, right?
Yes.
Was that true in all of the layouts, or the species compositions at least?
Yeah, that would not be true in every, if I were to actually calculate a difference between years for every single plot, it wouldn't have gone up for every single plot.
Well, not just every plot, I mean just on the trend anyway. So each treatment, each like the all grass, did you see a lift in
yes.
SOC in every one of them?
Yes, I did, sorry.
Why is that? Is that better than what was there before? Why, I mean, what happened? Why would that effect come out, and what can you say about that?
So if we go back, oh goodness, let's see, this figure. If we go back to this figure, right, the effect is, I told you to ignore this, but the effect is positive, right? So we do actually have a significant slope with richness, and this is a positive effect of every single one of these community types on soil organic carbon, right? So it would have happened in every single one of these species. And then I guess the next part was like, when I broke down the paths through which that could happen, that was me trying to make sense, because I've measured so many of those paths. It was making sense of why this would occur, because we wouldn't necessarily expect it.
Right, I guess, yeah.
I was going to follow up that too. I think initial conditions of the site or soil would have a big impact. In that area where that experiment was at was crop field, not all that long ago.
It was crop field, but we also mixed that crop soil with soils from Welda. So to facilitate the establishment of hard to establish plants, we were able to collect soils from a road extension project in Welda. And those soils had high organic matter because they were native undisturbed tall grass prairies that got mixed in. And it wasn't a small amount. It was like 75 gallons per plot that got mixed in. So our initial soil conditions actually weren't carbon deplete in top soils.
And kind of related in your first part of your experiment too, looking at the soil beds, you had different types of grasslands. I kind of wondered what you meant by grassland, because it seemed like they weren't all like native soil.
Oh yeah, no, they wouldn't
right?
be.
So to me, there'd be a lot of difference in a planted, well-managed grassland than a native prairie.
Yes.
Which would maybe provide different results.
Yes, it would. Yeah, and that's probably like the next step, is we have to actually partition and dig into some of those relationships and start exploring why we're seeing patterns in forested systems and not in grasslands, and think about what might be driving that.
So just to follow up then, so how long ago, or how long has that been transitioned back to prairie?
The dimensional
site where
sites.
you did the rain out
It
shelter.
is in my appendix. I think the last time it was prairie, I think it's been transitioning back since at least the 70s. It's been over 50 years.
It was probably till originally in 1880s or 90s, and then it was planted drum grass. I remember it was wheat, but it was planted drum grass.
And then the prairie grasses started accumulating because of the rodent exclusion code, because they couldn't mow it. And so just next to it, they were mowing it regularly, and that state brome, fescue, and this one, that area started being dominated by the prairie grasses
Yeah.
to the mowing frequency.
With that, can you assume that there was a flat baseline in SOC, or that it might have still had a slight upward trend as it was?
So, I mean, we do have the SOC concentrations in the first year, and there's no difference across richness treatments or like planted richness. There is a difference in subblocks. So there's some spatial heterogeneity that I account for in all of my models.
I mean, let's say you had a testing site just outside of your shelters that you didn't manipulate, but you just took soil organic carbon readings. Would you expect that there have been maybe a slight trend in that too, or is that pretty much, you think that's leveled out by now?
You mean like taking it over time? Do I think
Well,
that's...
just tracking as a kind of a control or a baseline for your experiment. What was happening right beside your experiment in the soil?
That is a good question. I don't know.
Because we've seen with no-till how quickly levels can bounce back, but that's, I think talking a couple of decades, and I don't know, but then they also start to plateau, so it could be very,
yeah, so one of the reasons that they tend to plateau is because you often saturate what they say is like the mineral-associated carbon, so the carbon that can bind to relatively small particles, right? That tends to saturate. One of the hypotheses for why grasslands accumulate so much carbon is because they start adding more particulate matter to the system, which doesn't get decomposed. So I guess one of the things that they often say is that molluscs are kind of like the exception to the physical limitation rule. Like sometimes you can get more carbon than you would expect in molluscs, but nobody really knows why. Like if you figure that out, nature. But I didn't take soil samples outside of the plots. And like I actually do have some not like plots that are not an experiment, but they still were mixed with the well-to soil. So I have plots that are outside of the experiment as like not necessarily control, but also like they're also practice plots. But they still were mixed with that well-to soil. So actually I don't have a good sense of what is going on like around. I guess we could try to get it. I will say that I would expect SOC to be relatively flat. They mow like very frequently. Well, we hope frequently to keep the snakes out. One person is nodding. Yes, they've been in the pots. Are there any other questions?
Thanks, Laura.